Dear students, in this today's video, we study the one part of genetics and plant breeding paper and point 3.3 maternal inheritance. In that maternal inheritance, we have to study what is mean by maternal inheritance, the difference which is present in between maternal inheritance and Mendelian inheritance, and we have to study this phenomenon with the help of a plastid inheritance. Before going to study first maternal inheritance, we study some basic information which is coming in that Mendelian inheritance. Inheritance of the most of the characters in eukaryotic organism shows the following characteristic features. Which are these following characteristic feature? In that first is nothing but in them the gametic, which is a gametic cell which is a produced, that is nothing but a male and female gametic cells which is a produced, and these gametic cells when these are unite, then it results into formation of a progeny. We say that F1 progeny, but here the one important thing is done, and that is nothing but a reciprocal crosses these are made. What is mean by reciprocal crosses? Meaning of reciprocal crosses means, for example, in a first parent, a first cross, we selected the male and female parent. These are having the specific phenotypic expression. Then in a first cross, if you selected male as a having white flower and female has a pink color flower, and in another cross these phenotypic character is a reverse, means female having male having blue flower and female having the pink flower, means the phenotypic expression of these parents, if these are changes, then that cross is termed as a reciprocal cross. And when such a reciprocal cross these are done, then in their F1 generation the progeny which is produced, these are having the identical phenotypic expression. Means when we study the Mendelian type of inheritance, in that all of you familiar the cross, that is a tall and dwarf. If you selected male as a tall and female as a dwarf, in F1 progeny we observed all progeny these are tall, but in a reciprocal cross, instead of a tall male, if you selected tall female and it is a cross with a dwarf male, in that cross also we observe the tall F1 progeny. Why this is thing we observe? Because in that the laws of a dominance is follows. Laws of a dominance means that reciprocal crosses. In both reciprocal crosses we observe the tall progeny, and that is seen in a Mendelian type of inheritance. This is the first point, that is the contribution by both male and female parents are equal, so that the results from reciprocal crosses are identical. But these reciprocal crosses give different result in case of a maternal inheritance. What is that we will study in a further plate. Then segregation produces the characteristic. These are produced in the ratio 3:1, and in the F2 generation we observe the ratio 9:2, 3:2, 3:1 in case of a dihybrid cross, and monohybrid cross that ratio is 3:1 we observe. These features of inheritance were first demonstrated by scientist Mendel, and therefore that a pattern of inheritance is referred as a Mendelian inheritance. It is universally accepted, and we all of you know that this Mendelian inheritance, for that Mendelian inheritance, the genes which is responsible, these genes, these are located only on the chromosome, and these chromosome present inside the nuclei. Inside the nuclear membrane, in the nucleus, that chromosome is present, and on that chromosome the genes these are present. Therefore Mendelian pattern is we say that it is a sufficient evidence for a gene to be located in the chromosome, and these genes these are termed as a nuclear gene, as well as we simply say at a gene. This is all about the Mendelian type of inheritance. But what is mean by non-Mendelian inheritance? In that non-Mendelian inheritance, we found that some characters in several organisms, they do not show this Mendelian inheritance, and therefore they do not follow the law of dominance of that Mendelian type of inheritance. The ratio 3:1, 9:3:2, 3:1, which is observed in monohybrid and dihybrid cross, these are non-seen in that non-Mendelian inheritance, and therefore this is referred as a non-Mendelian inheritance. In that non-Mendelian inheritance, we found that when we take that reciprocal crosses, then in that reciprocal crosses, the identical results we not observed. When identical results these are not seen in their F1 progeny, we say that it is a non-Mendelian type of inheritance. Again, in that case, we observe only the phenotypic character of a female parent. Only in that cross, this a female parent character only we seen in that a progeny, F1 progeny as well as a hybrid progeny. And when this is seen, the second condition, that is a female character is seen in that a progeny, we say that it is a non-Mendelian inheritance. And the third important thing is that when that same male and female is selected as a parent, and in between them in a future generations these are produced, means from that same, the F1 which is the progeny is produced, so the F1 character is kept as a male and it is a cross with the female. Then in F2 generation also female character we observe, or female parent character we observe F2. Again that F2 is remain as it is a male and it is a cross with the female parent. In that F3 generation also we observe that a female character, female parent character. When these are three conditions these are satisfied by that cross, then we can definitely say that it is a non-Mendelian inheritance. Again, in addition to these cases of non-Mendelian inheritance, some character in several organisms they shows the Mendelian inheritance pattern, but in that Mendelian pattern we observe, but individual is markedly affected by the genotype of that maternal parent of the concerned individual, and such cases this is termed as a maternal effect. Maternal means only the mother character, maternal, this term is related with that mother character, and therefore that mother character is seen, therefore it is termed as a maternal effect. The evidences for cytoplasmic inheritance, these are first presented by scientist Corens in a Mirabilis jalapa. Simply we say that a 4 o'clock plant in a 1908, and in them he observed there is a non-Mendelian inheritance which is termed as a cytoplasmic inheritance. Means only one character of these two parents, only the female parent they can be transmit its character towards the progeny, and as a result reciprocal crosses when we take in them they show the differences in the phenotypic character of their F1 progeny, and there is a lack of the segregation in the F2 generation as well as the subsequent generation, and therefore such a type of inheritance is referred as extranuclear inheritance, extra chromosomal inheritance. It is termed as a cytoplasmic inheritance, or it is termed as a maternal inheritance. Then again a question arises in our mind, if these genes they do not obey the laws of a Mendelian inheritance, they does not follow their laws, then which laws these genes these are follows, where these genes these are present, and how they transmitted their character generation after generation. For these questions the answer is in this manner. When these genes these governing the trait of a that trait showing the cytoplasmic inheritance, then these genes these are not present inside the nucleus, but these are present outside the nucleus, means these are present in the cytoplasm. Therefore these genes these are termed as a plasma gene, cytoplasmic gene, cytogene. Extranuclear gene means it is a present outside the nucleus, therefore it is termed as extranuclear genes, or it is termed as extra chromosomal genes, means these genes these are not located on the nuclear chromosome, therefore it is termed as extra chromosomal genes. and the sum total of gene which is present in the cytoplasm of cell, it is totally known as a plasmon, while all the genes present in the plastid, plastid means chloroplast, inside the cell organelle chloroplast, certain genes these are present, these are termed as a plastron. Means it is found that the inheritance of the most of the characters of an individual is governed by nuclear genes, no doubt. But in some cases, their inheritance means transmission of character generation after generation is governed by certain cytoplasmic factor or cytoplasmic gene or extranuclear gene. And when these transmission of character from parents to offsprings, these are governed by that cytoplasmic gene, it is known as a cytoplasmic inheritance, or it is termed as extranuclear inheritance, or it is termed as extra chromosomal inheritance. It is also known as a non-Mendelian inheritance, and it is also known as organeller inheritance. But again a question, why it is termed as organeller inheritance? In that process, the two organelles, these are playing important role which is present in the cytoplasm of cell. One is nothing but a chloroplast and another one is termed as a mitochondria. These are chloroplast and mitochondria, these cell organelles, these are having their own genome, and for doing any functions these are not depending on that nuclear gene. As these are having their own genome, own gene, they can do their function independently. Therefore, it is termed as a extranuclear inheritance, organelle inheritance, because in the organelle mitochondria and chloroplast such a gene is present, and that gene does the functions. They control that specific character independently, and therefore it is termed as a extranuclear inheritance. And that extranuclear inheritance, cytoplasmic inheritance, non-Mendelian inheritance, the example of a plastid inheritance means chloroplast inheritance and mitochondrial inheritance, these are coming. Again, we have to study the difference which is present in between that cytoplasmic DNA and a nuclear DNA. What is the difference? The cytoplasmic DNA is found in only chloroplast and mitochondria, while a nuclear DNA is found only on the chromosomes. Cytoplasmic DNA is circular except in ciliated protozoa. In that ciliated protozoa, their shape is linear one. While nuclear DNA, no doubt it is a linear one in case of a eukaryotic cell, and in case of a prokaryotic cells, these are a circular one. Then, in the cytoplasmic DNA, their synthesis is a continuously takes place throughout the cell cycle, while in case of a nuclear DNA, their synthesis occur only during the interphase of their stage. Cytoplasmic DNA replicates in both chloroplast as well as the mitochondria. When the replication of that cell organelle takes place, at that time the DNA which is present inside their replication takes place. And nuclear DNA, it is also replicates along with the chromosome. This is the basic difference which is present in between cytoplasmic DNA and nuclear DNA. Then question arises, that is why only the maternal character is seen in their progeny, only why mother character is seen by that progeny. Here, for that explanation, this chart is given to you or shown to you. This is a female parent and this is a male parent. During these male parent, these are female parent having the certain cytoplasmic content, and in the center nucleus is present. Male also having cytoplasmic content and nucleus. From that female parent, we observe the female gametic cells. For example, egg cells these are produced. At that time, the egg cell receive no doubt the daughter nucleus as well as the cytoplasmic content. Also, this daughter cell receives from their parental cells. But during formation of a male gametic cells, in this diagrammatic representation, we found that the male gametic receive only the nuclear material. It does not receive any cytoplasmic content from that male parent. And therefore, when union takes place in between that gametic cells, male and female, we found that In them the character which is present in that new one, it is only similar to their female parent, it is not similar to their male parent, and therefore this is a basic that is a things basic principle we say that due to which this cytoplasmic inheritance we observe. Means during the formation of a gametic cells from female parent, the egg cell which is produced, it receives both nuclear material as well as the cytoplasmic material from their parent cell, female parent. While during the formation of a male gametic cells, male gamete means for example sperm cells does not receive the cytoplasmic content from their parent cells, only it receives the daughter nucleus. Nuclear part, half nuclear part that gametic cell is received, and therefore when the union is that takes place in between such a female cells, gametic cells, and male gametic cells, no doubt their nuclear division fusion is that takes place. Both nuclear material is coming in that nucleus, but in the cytoplasm only the female cytoplasm is seen, and as it containing the female cytoplasm, therefore what happened, it shows the female phenotypic expression, and at that time we say that it is a case of the cytoplasmic inheritance. In this another cross, neighbor cross, we found that instead of a female character, correct, that female character is of a male and male character is of a female, therefore that cross is termed as a reciprocal cross, and these reciprocal crosses give result, means here female it is having the different characteristic, only it receive the female, and male cells receive only nuclei, and the union in between these takes place, and therefore the progeny which is produced in the first cross is a variegated one, and in the second these reciprocal crosses it is a green one. Means F1 get a different result in this cross. First cross the F1 progeny, these are variegated one, and in this grass the F1 progeny is a green one. Green character is of a female parent, variegated character is of a female parent. Means our three condition, first is nothing but a reciprocal crosses give different result, this is the satisfy. Second, in both female character is seen, that is a variegated character of a female here, and here green character of that female, these both character is shown by that hybrid progeny, and that character is transmitted generation after generation only through the cytoplasm of that female parent, and therefore we say that it is a case of a cytoplasmic inheritance, extranuclear inheritance, maternal inheritance. The first case of a cytoplasmic reticulum, cytoplasmic inheritance, all of you know that it is studied by scientist Corens and in Mirabilis Jalapa, and he observed that the basic difference between that Mendelian inheritance and cytoplasmic inheritance in this manner, again the on the basis of the genes, nuclear gene, plasma gene, segregation pattern in Mendelian inheritance is a distinct one. In case of a cytoplasmic inheritance, it is not a distinct one. In Mendelian inheritance, reciprocal differences these are not observed, but cytoplasmic inheritance, in them we observe the different F1 results. In Mendelian inheritance, we does not see the maternal effects, but that maternal effect is present in cytoplasmic inheritance. Gene mapping in case of a Mendelian inheritance, it is become easy one, while such gene mapping is become difficult in case of a cytoplasmic inheritance, and the location of a gene in case of a Mendelian inheritance only on the chromosome. That chromosome is present inside the nucleus, while in case of a cytoplasmic inheritance, these genes are located on chloroplast or mitochondria. This is all about the basic things present behind the cytoplasmic inheritance. Then our next point, how that a cytoplasmic inheritance is takes place, we have to study with the help of that plastid inheritance. And that plastid inheritance, for that we selected the example of a Mirabilis. Mirabilis jalapa means it is a 4 o'clock plant. and that inheritance in a plastid characteristic is seen due to the gene plasma genes which is located in a plastid. Plastid inheritance was the first described by scientist Koren in 1908 in the 4 o'clock plant. Here, when we observe that Mirabilis jalapa, we found that the leaves, these are having green patches, white patches, as well as a mixing of that both patches, and therefore it is termed as a variegated one. Some branches of these plants are having only green leaves, some are having only white leaves, as well as some branches, these are variegated leaves. What is mean by variegated leaves? The variegated leaves means it is having the mixing of white and yellow spot, and these are again a variable size, and these are present on the green background of a leaf. Therefore, it is termed as a variegated leaves. Means here same plant, that is Mirabilis jalapa, some branches these are having all green leaves, one branch these are having the variegated leaves, while one branch these are having all white leaves. Here you remember well, the green branch, these are having the green leaves because in them green color chloroplasts are present. Variegated one means there is a patches of white and yellow leaves, correct? And that is a variegated one. And white one means there is a lack of a chlorophyll pigment. But as there is a lack of a chlorophyll pigment, that branch or that on that branch the leaves which is a present white color, they does not take part in the process of a photosynthesis, and therefore they does not produce their food material. But these are plant other branches which is a green one, they prepare food material and these are supplied to that branch. Therefore, these white leaves, these are seen on that plant. Same situation in case of variegated plant, where green patches these are present, means in that region the chloroplast is present, therefore it is take parts into the photosynthesis. Here Korean scientist, he does the reciprocal crosses. In the reciprocal crosses, first he selected female parent as a green one and male parent as a variegated one. Then in the F1 he observed all these are green offsprings which is having the mother character. He selected the reciprocal cross, means instead of a variegated plant as a male, he selected as a male green one and female variegated one. From that reciprocal cross, he observed the white offsprings. Means here, first condition is satisfied, the F1 results is a different one. Reciprocal crosses gave different result. In first cross, green offspring is present. In another cross, white offspring is present. As this white has offspring, they does not having the chloroplast, they do not produce food material and therefore they cannot remain alive longer time. But when that green offsprings, these are maintained as a male, maintained as a female in every cross, and it is a cross with that white offsprings. White offspring is selected as a female. Then in a further progeny, we obtain that the green plants these are produced. Means that reciprocal crosses gave different result. In the reciprocal crosses, maternal character is seen, and that maternal cross, if it is maintained in each and every generation as a female parent, then in a further generation, we observe that a female parent is remain a constant in a further. Therefore, it is a case of a plastid inheritance. It is a case of a cytoplasmic inheritance. It is not a case of a Mendelian type of inheritance. And therefore, these scientists, he does the crosses in between green, 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 white, green variegated, and in them the progeny which is produced, these are green one. When he selected white female and male green, white variegated, in all he observed the white progeny. When he crosses variegated with again these three green, white, variegated, then in that case green, white, variegated in various ratio in each crosses these are produced. Means when he selected a female as a green, white, variegated, and it is a cross with these three types of the male, then only the female character is seen in that F1. Here the female character white, therefore female character is seen. In this variegated one also, in various ratios, each character we observe, correct? Means this is nothing but a mother character is seen. Maternal character is seen in case of a mirabilis jalapa. In this diagrammatic representation also same case, that is which are the male parent, which are the female parent, from them the female character is transmitted and it is seen in that F1 progeny. This is shown in this diagrammatic representation also. And therefore, it is found that from these various crosses of leaf phenotypes of mirabilis jalapa, it clearly indicates that leaf phenotype of a progeny is the same as that of the female parent. And these phenotype of a male parent do not contribute anything in that progeny which is developed. Therefore, it is a case of only and only cytoplasmic inheritance. The genes which is responsible for that leaf phenotype, these are only transmitted through the cytoplasm of a mother parent. Cytoplasm of a mother parent, and it is therefore termed as a cytoplasmic inheritance or extranuclear inheritance or maternal inheritance, because for that character the genes which is present, these are present only in the chloroplast, only in the plastid, and that plasmogenes they control this character phenotype of that leaves. In case of a mirabilis jalapa means 4 o'clock plant, की जे प्लांट हे गणपतीला आवडतं, ज्याला आपण गुलबक्षी मराठीत म्हणतो, ज्याची फुलं 4 वाजता उमलतात आणि ती फुलं वेगवेगळ्या प्रकारची गुलाबी, रेड, पिंक कलरची आपल्याला फुलं ती बघायला मिळतं. आपण लहानपणी ती फुलं गोळा करून आणतो, त्याचं असं गुंफतो त्याचे सीडचे पोर्शन आणि अशी वेणी हार करून आपण गणपतीला घालतो. दॅट इज टर्म एज अ 4 o'clock mirabilis jalapa. In them, we observe the cytoplasmic inheritance. Thank you, students.